welcome to the another of the Classics of Immunology Journal Clubs. And before we get started on this particular one, I want to remind you to like it on YouTube, on the YouTube site. We're getting more and more. It's just really good. I'm happy about that. And then click on the bell so you get notified. And then also scroll down on the YouTube site that you can access the paper that we're going to review, as well as go on down to my own website. And on my own website, kendallasmith.com, you can go on the writing tab at the top. Then you can access all of the papers, the classics of immunology. So today we are already up to 1980. It's amazing. And we're going to review a paper by Hussey Reinherz, Rebecca Hussey, and Stuart Schlossman, entitled "A Monoclonal Antibody Blocking Human T-Cell Function." And just to give you a little background into these people, we talked about Hussey Reinherz last week. At this particular point in time in his career, he was a postdoc with Stu Schlossman, and that's why Stu's the senior author on this paper. Rebecca Hussey was a technician who was tremendously gifted in lymphocyte culture. Stu Schlossman was a product of NYU in New York City for his undergrad as well as his medical school. While he was there, he got under the influence of Baruch Ben-Asaraf, and we've talked about him a little bit in the past. He went on there from after he graduated from medical school, and he did a postdoc in immunology at Columbia, and then went out to WashU in St. Louis and did a fellowship in hematology. And then he came back to the East Coast, and he got his first job with Beth Israel in Boston in the Harvard complex. And he was interested in trying to differentiate different kinds of lymphocytes through any means possible. And this came at him from hematology. He was interested in trying to identify different kinds of lymphocytic leukemias and so forth, as was Ellis at this stage, or earlier in his career. And Ellis, he was, maybe we didn't talk about him the last time. We did, but now we're going to talk about it. He was a product of inbred Boston, really. He went to Harvard College, went to Harvard Medical School. He did his internship and residency with a combination of Mass General and the Peter Bent Brigham. He then went on to doing a fellowship at the Dana-Farber, and it was in hematology and oncology. And he told me, Ellis is a good friend of mine, so I know a little bit more about him than some of these other people that have written papers that we've discussed. He told me that when he was on rounds and they were discussing the leukemias and the lymphocytic leukemias and so forth, he was just so dissatisfied with the quality of his mentors in hematology, and he thought that they were completely bereft, and so he just left hematology. And so he took a research postdoc with Stu Schlossman. So in the introduction of this paper, so this is about a monoclonal antibody that blocks human T cell function, which is the title. And in the introduction of this paper, he states right off that what everybody knew, and that was, although we know what the antigen receptor is on B cells, and that's surface immunoglobulin or antibody, we don't know what the nature of the T cell antigen receptor is, and that's where he was focused. And they got together with a group at the Ortho Pharmaceuticals down in New Jersey. And this is a collaborative effort, and I'll get into what each of these people did. And together, they made a series of antibodies, monoclonal antibodies, that were reactive to human T cells. And this was the beginning of really a flip-flop of where the real immunologists up until this time had been focused on mouse cells, lymphocytes, and so forth. And as you know, the Kohler and Milstein were able to show that you could get mouse monoclonal antibodies by immunizing mice with different antigens and then doing fusions with mouse plasmacytoma cells. Well, that made it a little difficult to get antibodies to mouse lymphocytes because they were mouse. And therefore the mice were basically tolerant of themselves, hopefully. And so the mice people were in a quandary. Meanwhile, there weren't very many people working with human lymphocytes. I was one of them. Ellis and Stu Schlossman were others. It was almost like, you're home free. You don't have all the competition of all these real immunologists up breathing down your neck. And so this is when the study of human T cells really took off, because what they did is that they made a series of antibodies, monoclonal antibodies that they called OKT1, T3, 4, 8, and so forth. And they'd done a series of papers in 1979 that I haven't reviewed here, but you can find them in the references to this paper. So if you're interested, you should read all those papers, or at least look at them, because of the fact that I'm surprised they had time to write them. They had so many papers on where they showed how they did how they made these antibodies, and then their initial characterizations of them. Now, this is about one of the antibodies of about a half a dozen or so that was focused on an antigen or a molecule on the surface of the T cell that was called T3. And when they looked through all of these antibodies to see what they would do, it was only anti-T3 that had any functional influence on the T cells at all, and it inhibited their proliferation and other things. And so on the basis of that, they went through a series of experiments that are in this paper. And in the introduction, they concluded that on the basis of what their findings were in this paper, that this T3 molecule on the surface of human T cells must have something to do with T cell antigen recognition. That's very important. That's 1980 in the human, and the mouse people were 5 years behind and really never caught up. In the materials and methods, they're pretty simple. The isolation of the lymphocytes, human lymphocytes from the peripheral blood. By this time, it started out with Peter Noel in 1960 that we reviewed. And so this is 20 years later. And by now you could take a mixture of a polysaccharide and a lipid glycol hypake, and layer the blood on top of that, and then spin it at a particular speed in the centrifuge. And the red blood cells would agglutinate and go to the bottom of the tube, and the lymphocytes would hang up right on the interface, right above the glycol hypake layer. And then you could just reach down with a Pasteur pipette and suck them off. And then you had you got rid of the granulocytes in that instance, which were 70% of the peripheral blood cell, white cells, whereas only 30% were mononuclear cells. So this concentrated the mononuclear cells. And then they further purified them by incubating them with sheep red blood cells, because it had been discovered at that time that the T cells, human T cells, would or the sheep red blood cells would agglutinate around the T cells, but would not do that to B cells. And so they called those because they could make cytospins and put them down onto a microscopic slide and stain them and look at them. And so you'd see a lymphocyte, and then you'd see all these red cells around them, and they looked like a rosette. So those were E rosette positive, and those were T cells, and the E rosette negative cells were everything else. They also purified macrophages by causing them to be just incubating them on plastic or glass for a while, and then washing off the non-adherent cells, and then scraping off the monocytes with a rubber policeman kind of thing. So that's how they got their cells. Their monoclonal antibodies, what Ellis described to me is that their lab was responsible for immunizing the mice. And then they would send the mice down to ortho, who had a flow cytometer. That's the other thing that this paper illustrates, that the power of the flow cytometer as a device to look at particularly fluorescent antibodies bound to different kinds of cells. And they would send the mice down to ortho in New Jersey. They did the fusion, and then would send the supernatants of the fused hybridoma cells back up to Harvard. Harvard would screen them then for antibodies that would recognize whatever. They just wanted to know what they were recognizing. E rosette positive, E rosette negative, macrophages, and so forth. So they assayed the functional aspect of this in this paper, the proliferation assays, whereby they would activate peripheral blood mononuclear cells in vitro, and with different kinds of stimuli. They used a mixed lymphocyte culture. They used soluble antigens, mumps virus, and tetanus toxoid protein antigen. And they would test them with and without the antibodies to see whether or not there was any influence on the proliferative capacity of these cells. They'd culture the cells for 5 days, and then they would pulse them overnight with tritiated thymidine. Then they also assayed for lymphocyte-mediated cytotoxicity in the standard way. It became standard by then. You remember back in '68, Serratini and Brunner and Serratini had described the chromium release assay. So you take your target cells, label them with chromium 51, incubate for a while, then wash off the excess, and that's your target cell population. Mix the cells then at different lymphocyte to target cell ratios, and then looked at incubated them for about 4 to 6 hours, and then harvested the cells, took the supernatant and assayed that in a radioactive counter for chromium 51. They also measured B cell function and the ability of activated human cells. It was known by then that you could use another mitogen for lymphocytes called pokeweed mitogen. Pokeweed mitogen activated both T cells and B cells, but in addition, unlike PHA or concanavalin A, pokeweed mitogen would cause the B cells to start to secrete immunoglobulin. And in order to do, and so they tested their antibodies, their anti-T cell antibodies, to see if it would influence that particular assay. And they would culture these cells, the whole mixture of cells, for 7 days, and then wash the cells and look for using a radio immunoassay, look for immunoglobulin in the soups of these cells. So, and the results, which I'm going to now show you by screen sharing. Okay, now this is on the top left-hand corner now is figure 1, which shows the unique capacity, as it's in the title there, of anti-T3 to inhibit MLC proliferation. And you see what you have is on the y-axis is tritiated thymidine incorporation in counts per minute, and then the control with no antibody added to the cultures versus anti-T1, T4, T5, T8, and it's a beta-2 microglobulin, an anti-IA, and anti-T3. This is medium alone. And so of all these antibodies, only anti-T3 blocked the proliferation. Quite striking. Then they thought, well, when do you have to add it? How long does the antibody have to be there? And so this is the time days anti-T3 was added to the MLC, which was allowed to go for 6 days, and then harvested. So if you delayed the addition of the antibody by as little as 1 day, it would only you'd only get 40% or about half as much proliferation on day 6. And by 2 days, it's almost all gone. And by 3 days, it's totally gone. So it was inhibiting something early on. Here's 4 hours early on in this whole mixed lymphocyte reaction. Question is, what would be that? So then they wanted to do a dose response. They were using, for the most of these experiments, they were using ascites from mice that had ascites because they had plasmacytoma hybridomas growing in them that were releasing huge quantities of antibodies. So here they've diluted out their anti-T3 from 1 to 100 up to 1 to 100,000, 10 to the minus 2 to 10 to the minus 5. Here the proliferative stimuli are listed. So MLCs in this column, tetanus, toxoid, and then mumps. And you can see that the ascites control, with MLC, we've got 116 counts per minute. With anti-T3 at 1 to 100 dilution, it's already down to 7,000. And you see the same general pattern. Then it dilutes out, but it stays around. Here's 1 to 10,000. So that means that you don't have too many other kinds of things in there except these antibodies, essentially. Then the tetanus, toxoid, and then mumps, same story here. Then in figure 2, and this they mentioned was a representative of 12 separate experiments, and they were all basically just like this. Then in figure 3 we're into now, where they decided that because they were using ascites, crude ascites for all these experiments, that they should use purified antibody. So they purified some of it, and they could dilute it out from 1 nanogram per 10 to the 5th cells, diluted out to 1 to 10,000 from that. And so this basically saying this is what's doing the trick. And then we go on to table 2, where they show that basically it's the anti-T3 antibody that inhibits the generation of cytotoxic T cells. So now they're doing an MLC. They're not measuring proliferation, but they're measuring it. They harvest the cells at the end of 5 or 6 days and then assay them for cytotoxic reactivity on the target cells. And you can see that basically this is the percent cytotoxicity at 20 to 1 lymphocyte, 10 to the 1, and 5 to 1. And when they added the monoclonal antibody at the initiation of the MLC, the other pan T cell antigen that they looked at was T1. And you can see that here we got the control. They have 31% cytotoxicity, and anti-T1 brings it down to 24. You can say whatever, but then looking at a 10 to 1 ratio, it was 23 is down to 14 and 16 down to 10. The anti-T3 was better. It brought it all the way down to 7 here against 30. But then the specificity controls, anti-T4, anti-T5, anti-IA, beta 2, there was no inhibition of the cytotoxicity. So it was in the generation of the cytotoxic T cells where this antibody early on seemed to be doing something to these things. And they decided, okay, well, let's look at the effect of the antibody activity on the actual cytotoxic reaction. So they let the cultures go for 5 or 6 days, harvested the attacker cells and put them into lymphocyte-mediated cytotoxicity assays. And at the same time, then adding the different antibodies, and none of the antibodies, even anti-T3, inhibited the cytotoxicity of these cultures. So that it wasn't. And one of the other things that they mentioned in the paper, the antibodies were not killing the T cells, but they certainly were inhibiting their function. And then the last thing one of the last things that they wanted to look at was the effect of the anti-T3 antibody on pokeweed mitogen stimulated immunoglobulin production. And so here is the one experiment they show us with immunoglobulin on the y-axis And T cells, purified T cells, purified B cells, you need both to get immunoglobulin production. And then if you add anti-T3 at 1 to 100 or 1 to 1,000 dilution, or even 1 to 10,000 dilution, you see that the pokeweed mitogen-induced immunoglobulin production is inhibited. The last thing that they talk about then is they did a timing experiment with the immunoglobulin production. And just like in the generation of the proliferative response and in the generation of the CTL response, you need to have the anti-T3 there early. If you wait, you're not going to be blocking the reaction. So I will stop screen sharing there. So that was it on that paper. And they had, so the data were really pretty clean and clear cut. And in the discussion, they make several points, like you do in these kinds of papers. They said, number one, anti-T3 blocks proliferation even at a very low concentration, 1 nanogram per million cells. It only really blocks well if you have it present in the first 24 hours after the activation step. And therefore, they conclude that there must be something about early antigen recognition or early activation of the T cell that this antibody is blocking. And as well, the anti-CD3 blocks the generation of cytotoxic cells as well as the generation of immunoglobulin formation. And again, only if it's there early within the first 24 hours. They go on to say that, because of all the different antibodies they tested are from different isotype classes, it's not specific for a particular isotype of immunoglobulin. And when they've looked at where the T3 surface molecule is expressed on, let's say, thymocytes and peripheral blood lymphocytes and so forth, it's only on 10% of the thymocytes that they looked at, of human thymocytes now we're talking about, whereas it's on all of the 100% of the peripheral T cells. And it's therefore they say, whatever this molecule is, it's appearing late during T cell ontogeny in terms of the maturation of a mature T cell in the thymus. They reiterate that the T3 effect, the anti-T3 effect, was not due to the fact that antibody was killing the cells. And they go on to say then that it may be that what this T3 molecule is an invariant region or chain of the T cell antigen receptor. That was sort of an earth-shaking comment to slip into their discussion part of this paper. But then they go on in the very last paragraph, they speculate that anti-T3 may be used to treat auto-inflammatory, autoimmunity diseases. And it also might be able to be used to treat organ transplants like kidney transplants, as well as bone marrow transplants. And in actuality, this paper came out in 1980. They submitted a patent for this paper, for this anti-T3 molecule in 1982, which was then licensed in 1984. And it was really the first therapeutic patent for a monoclonal antibody that had been awarded. One of the things, and then subsequently, that we know about all this sort of thing, I don't want to give away too many secrets at this stage of the game, but when they used anti-T3 in the clinic in humans, they found that they would activate the T cells, all the T cells, or at least seemed like it activated all the T cells. And you could get the cytokine syndrome from that. That was a precursor to what we're dealing with now in 2021 in the cytokine syndrome story of chimeric antigen receptor T cells and so forth. They speculated or concluded that the T3 molecule could very well be the signaling component of the T cell antigen receptor. And then they went on subsequently just after this to show that this antibody would cause a rapid increase in intracellular calcium that happened within a few minutes of the addition of the binding of the anti-T3 molecule onto the cell surface. So that's just the illustration. The mice people did not recapitulate this whole story for 4 or 5 years with anti-T3. And as we'll see, as we go on in the Classics of Immunology Journal Club, Reinherz, they were on a roll up there in Boston, and they were the first ones one of the first ones to make human T cell clones. They made cytotoxic T cells out of CD4 positive cells as well as CD8 positive cells. And that's interesting. And we'll get to that eventually. So please like us on the YouTube channel, click on the bell, access these papers, these classic papers, and read them through. Now that we've gone through a little bit of this, you'll learn a lot. Thanks so much.